Today's lecture is on variations in a chromosomal structure. Variation in a chromosome morphology or structure. The chromosomes are the structures with a definite organizations. They carry the genes which remains arranged in them in a linear manner. In orderly conditions, the chromosomes remains unchanged, but under certain natural or artificial conditions, some structural changes occurs in them. These structural changes are collectively called as chromosomal aberrations. Most chromosomal aberrations are caused due to accidental, natural, or induced breakage, may be caused by radiations and chemicals such as lysergic acid or diethylamide. The chromosomal aberrations may occur in a single chromosome or may extend to both of the members of pair or may involve two or more pairs of chromosomes. The chromosomal aberrations may be of following two types. First, that is the intrachromosomal aberrations, and second one, that is the interchromosomal aberrations. First of all, we will see the intrachromosomal aberrations. It occurs in a one and the same chromosome. It is of two types. First, that is the deficiency or a deletion. When you observe the picture, when the chromosome lacks in a intracalary or terminal segments, the deficiency or a deletion occurs. The last chromosomal segment, if lacks in a centromere, lag in anaphasic movements, and lost from a recognizing nuclei. A loss of any considerable portion of chromosome is usually lethal to diploid organisms because of genetic imbalance. Then another one, that is the duplication. It occurs when a segment of the chromosome is represented two or more times in a chromosomes. The extra segment may be attached to the chromosome when a loci are repeated or to a different linkage groups or even be present at a separate fragment. The duplication of chromosomes protect the organism from that effect. It is useful in a evolution of new genetic material and in altering the phenotype due to reallocation of chromosomal material. Then another variation in chromosome structure, that is the inversion. An inversion is the chromosomal aberration in which a segment is inverted at a 180 degree. Inversions are called as pericentric when the segment includes the centromere, and a paracentric if the centromere is located outside the segment. When a crossing over occurs within the inverted segment of paracentric inversion, dicentric and acentric chromatids are formed. The dicentric chromatids form a bridge that breaks when the anaphase chromosomes separate towards the pole. If crossing over within a loop of pericentric inversion, the chromatids produced with a deficiency and a duplication. Then we will see the another type, that is the interchromosomal aberrations. When a break occurs in a non-homologous and resulting fragments are interchanged by both of non-homologous chromosomes, the interchromosomal aberrations occurs. The interchromosomal aberrations includes following types, that is the translocation. It involves the shifting of part of one chromosome to another, the non-homologous chromosome. If two non-homologous chromosomes exchange parts which needs not to be same size, the result is a reciprocal translocation. In a reciprocal translocation, there are two types, that is the heterozygous translocation and homozygous translocation. In a heterozygous translocation, one pair is interchanged, one pair is a normal one, while in a homozygous translocation, both the pairs, these are interchanged one. In a homozygous normal, both the pairs are normal, but in a heterozygous translocation, one pair interchanged and one pair normal. while in a homozygous translocation, both the pairs are interchanged. Reciprocal translocation is of two types. It is the heterozygotic translocation and homozygotic translocation. In a heterozygotic translocation, considerable degree of meiotic irregularities occurs. Typically, cross-shaped formation is seen in a prophase first. This often open out into a ring as a chiasmatic terminalization. It produces semi-sterile organisms because half and two-thirds of the gametes or meiospores fails to receive the full complements of the genes required for normal development of sex. It is a common in maize plant. While in a homozygotic translocation, in this type, the normal meiosis occurs and that cannot be detected cytologically. Genetically, they are marked by altered linkage groups and by fact that a gene with new neighbors may produce a somewhat different effect in its new location.